My name is Marty Schiller. I'm the Executive Director of the Nevada Institute of Personalized Medicine at UNLV. And this product I'm about to discuss came out of my lab. And the product is called the VCSOnco. And it's a way to enable personalized medicine in the world. Next slide. So I want you to think for a second about yourself. And either you or someone in your family has probably had cancer. And if that's not true, you've had a friend. And I don't even have to ask you because I know it's true because the frequency in the world is enormous. And often it's deadly. So humanity has to live with this scourge, and you do too. And there's no way to solve it now. One of the best inroads into this in the past decade is from personalized medicine, where we can sequence the genome of the cancer cell and then identify what drug should be used. And it's showing huge effects already. This year, cancer deaths for the first time in decades are down. But what you don't know is the limitation. A gene, a typical gene, can have 20,000 different mutations. And when they do a diagnostic test, the most they're doing is a few thousand. Often it's hundreds. So you're getting tested and told you're negative for cancer. And you're not even being really tested. You're just being tested for a few known positive mutations. This limits the entirety of personalized medicine. There's 300 cancer genes. And when you get tested, you're only getting tested for a few, and they really don't know. But the test does work sometimes. The fact that it works so much, it's such an opportunity to improve the lifespan of humanity. Next slide. So what we're going to do are create these gene atlases of mutation. My laboratory's developed a technology where we can take a gene and study it in a test tube, and we can figure out what all 10 to 30,000 mutations do, so that you have a way of interpreting the genome when someone comes in with cancer. Okay. That's not even close to an ability that any laboratory has in the country at this point. Okay. These variants are called variants of uncertain significance because we don't know what they do, and that they're given a name. And anytime you run a test on someone, a genetic test, they have the USs, and they're ignored. They're not even reported. Our company is named Variants of Certain Significance because we solve this problem. I just sent out a grant for review earlier in the year, and I got this report back from a reviewer. And these reviewers on these NIH grants are harsh. His comment was, I can think of no larger impact on healthcare than being able to determine the clinical significance of the USs. So this is a third-party verification. Next slide. Okay. So we estimate that each gene variant map we produce will be worth millions of dollars. And if we consider just 5 of the 291 known cancer genes that are the best studied, they only have 5% of the mutations mapped. So for you, you come in, there's your genome, all those spots are potential mutations that might cause cancer. But when they run their tests today, you're only getting a few of them. That's what's going on in the world. And it's routine, and any expert will verify that. Next slide. So our solution is what we've done is the way they look at these today is in the best studied cancer gene called BRCA1. Myriad Genetics has been studying this gene for 30 years. And today they know what 5,000 of the 38,000 mutations do in that gene. The rest they don't know. Okay. What we've done is we've reduced the time where we can today do 50,000 mutants in 3 months, but we're optimizing, and this is increasing rapidly, and we're expecting, based on some calculations, we'll be at 15 million mutants in a 3-month period within a year of R&D. But today's capability is just fine to analyze genes. Our product is a B2B license of these maps for these cancer genes. We call them GAMs, or Gene Atlas Mutations. We don't require FDA approval because we're doing this B2B, and we can do multiple products per gene. There's different regions of the gene called the promoter, coding region, alternatively spliced exons, and then we can do single mutations, or our throughput is so high, we can do double mutations, which are often observed in patients. Next slide. So who are we going to be licensing this to, and what is the approximate markets? So we see 6 areas for licensing to existing companies. Each of them have dozens of companies, and they're multi-billion-dollar industries. So cancer testing and treatment, drug discovery, predictive screening for newborns, carrier screening for mutations, nutrigenetic testing, and there's something called the Cancer Research Moonshot Project. The market for 2017 in these areas is $2.6 billion, and the projections are there. The total market for cancer treatment is $100 billion per year. Next slide. So we have 291 GAMs, and we have 6 products per GAM for a total of 1,455 products. Considering 6 licensing opportunities per product, that gives us about 8,900 licensing opportunities, and we think a conservative estimate on the licensing opportunity for each GAM based on the markets is $3 million. So we think our target opportunity, this is an old slide, is $26 billion. The data life is extensive, and once it's developed, there's no value to duplicate it. So we're going to take your chromosomes and take one gene out at a time and process it into these maps where you'll know the potential to cause cancer for each potential mutation. Next slide. Our product timeline is here. We think in the first year that we can produce 2. That's because we have to set up the laboratory. In year 2, conservatively, we can do another 2, and then we scale it over the first few years. So because we can make so much money from each GAM, the company quickly becomes self-funding and should have a very large growth. Even with the staff that we start with and the facility, we should be able to scale to easily do 50 to 100 of these per year once we get going in the company. Next slide. So we have a really unique competitive advantage that our GigaAssay technology is the only means to do this. We are the only company that would be doing the promoter portion of the genes, and we're the only company that would have the capability of doing double mutants. The B2B customer who licenses our data really should dominate their market quickly because they're going to have such a superior product to what's currently being sold. So no more surgery from inferred incorrect synonymous mutations, which is going on. Say goodbye to VUSs and hello to VCS, variants of certain significance. Next slide, please. So what does the competitive landscape look like for people that actually have known variants? There's only one company. It's Myriad. They've been doing this for 25 years, and I just gave you a summary of their data. They're really far away from what we're going to be capable to do in the month time frame. There's a public database that everyone else uses. It's called ClinVar. But both of these have enormous disadvantages, and I won't go through them all, but if you want to, I'll come back to them later. But we have a number of advantages over the existing company. Next slide, please. So we told you that we're going to be licensing to B2B customers in different areas. Here's just a picture of some of the companies. Pharmaceutical companies for drug development. I didn't mention that one before. Cancer research, carrier screening, genetic testing. I've been through the list. We have multiple products. Our approach is going to be to produce the product and go to that field and have sealed bid auctions. We think that will maximize value of our products in this way. There's also something called the Genetic Test Registry by the National Center of Biotechnology Information. And if you go into their database, there already exists 11,000 conditions, 4,500 genes and 500 laboratories. So there's ample room for expansion of a further product line with our technology. Next slide. Our financial summary that we're projecting is we have the revenue and profit that's basically shown here. You can see how it basically scales. And this is based on the number of products I presented in the earlier slide and the assumption of $3 million per product. And that's based on sales of similar products to these companies in these industries. Next slide. I'm Martin Schiller. I'm the CEO. I have 21 years of experience as a research scientist. I've run a research laboratory, and I founded and I'm running a research institute. So I have experience in managing all of the necessities for running a business. I also have started up companies previously, never one in my own space, so I'm excited about this opportunity. I'm co-inventor of the GIG assay, and I've been a professor at Johns Hopkins University, University of Connecticut Health Center, and now at UNLV. We also have Jim Timmons, who's on board, who's serving the role of a chief operating officer, and Dana Ono, who's run a venture fund and has 30 years experience in the Boston area in the biotech sector, who's serving as a sales broker and advisor, has worked with the seal bid mechanism before. Next slide. So what's the current status? We've proven the technology. My laboratory has obtained about $1 million worth of funding. And what we've done is we used it on a single gene called TAP and showed that the technology works. We engineered software to analyze the data and completed that. And then we've gotten an exclusive license from the university to practice the technology. The next steps are to build a lab and start cranking out genes. Next slide. So why should you invest in our company? Time is right. This is a growing market. Genetics is an important space today. We have a proven proprietary platform technology with a deep product line. Our technology is going to be transformative in several ways that I've discussed. We have an experienced and capable team. And then our model is B2B licensing and perhaps direct-to-consumer sales of these GAMs later on into large markets. Our call to action is we're seeking $2 million to run the sales and marketing operations of the company, and this company will be purchasing or licensing these GAMs from another company called Heligenics that I'm also involved in. Next slide. So I have a bunch of backup slides to answer questions, but thank you for your time, and I'm happy to answer any questions.